Hey everyone, welcome back. So let's jump right into today's explainer. We're going to be exploring the totally invisible, microscopic world of enzymes. If you've ever wanted to understand how life itself is actually possible down at the chemical level, well, you're in the exact right place.
Just think about this for a second. Right now, as you're taking this in, your body is performing literally millions of incredibly complex chemical reactions. If you tried to pull off these same reactions in a chemistry lab, you'd usually need extreme heat, massive pressure, some highly volatile chemicals, the whole shebang. Yet your body just cruises along doing it all at a perfectly comfortable 37 degrees Celsius. So how are you not spontaneously combusting right now? What's the biological problem solver keeping everything running smoothly? Well, the answer is enzymes.
Okay, let's look at our roadmap for this explainer. First up, we'll meet the biological catalysts. Then, we'll cover how enzymes actually bind to substrates. From there, we'll look at lowering the energy barrier, followed by our main event, factors affecting enzyme action, and finally, measuring the invisible. All right, part 1, meet the biological catalysts. So at their core, enzymes are biological catalysts. They're biological because, of course, they work inside living systems, and they're catalysts because they speed up the rate of chemical reactions. And here's the kicker. They do this without actually being used up or permanently changed themselves. Structurally, they are globular proteins, which just means they've got this very complex folded 3D shape. And I really want to emphasize how absolutely crucial this is. Virtually every single metabolic reaction in any living organism is controlled by an enzyme in some kind of biochemical cascade. Without them, those chemical reactions keeping you alive would just happen way, way too slowly. So yeah, they are literally essential for life to exist at all. Now, enzymes don't just hang out in one specific place. We've actually got both intracellular and extracellular enzymes. Take intracellular enzymes, for example. These are produced and do their job right inside the cell. A really perfect example of this is catalase. Your cells are constantly producing hydrogen peroxide as a metabolic byproduct, which is highly toxic. It's bad news. But catalase swoops in and incredibly fast converts that toxic peroxide into totally harmless water and oxygen, literally saving your cells from destruction. On the flip side, we have extracellular enzymes. These are secreted by cells to do their heavy lifting on the outside. Digestion is a great example here. Macromolecules of food are just way too huge to enter your cells directly, so enzymes like amylase are secreted out into the gut to chop up those large carbohydrates into simple sugars that your body can actually absorb and use. Moving right along to part 2, how enzymes bind substrates. So if we zoom way in, every enzyme has a very specific little cutout. We call this the active site. And this active site has a highly specific shape that's perfectly designed to fit a very specific substrate. The substrate just being the molecule that the enzyme is actually acting upon. But, it's not like they just magnetically snap together from across the room. It's a game of chance. The substrate literally has to physically collide with the enzyme's active site at the exact right orientation and at the exact right speed for a reaction to even trigger. When they do finally align perfectly, they form what's known as a temporary enzyme-substrate complex. Now, for a really long time, scientists thought this whole interaction was just like a rigid key sliding into a stiff padlock, the classic lock and key hypothesis. But science has actually updated that view. Today, we understand it through what's called the induced fit model. Basically, instead of being completely rigid, the enzyme and its active site actually change shape slightly as that substrate molecule enters. We call these conformational changes. It's almost like the enzyme is giving the substrate a little hug. This ensures an absolutely ideal binding arrangement, maximizing the enzyme's ability to actually catalyze the reaction.
Which brings us to part 3, lowering the energy barrier.
Okay, so every chemical reaction out there requires a certain amount of upfront energy just to get the ball rolling. We call this the activation energy. Normally, without an enzyme, you have to pump a massive amount of energy into the system just to make the substrate unstable enough to react. But when you bring in a catalyst, like our trusty enzyme, that activation energy barrier drops dramatically. It's a total game changer. But here is the really crucial takeaway. The enzyme isn't actually adding new energy into the system. Instead, it's providing a completely alternative energy pathway entirely. Remember that little induced fit hug we just talked about? Well, when the enzyme does that, it physically influences the stability of the bonds inside the substrates. It destabilizes those bonds, making the substrate far, far more reactive than it ever would be on its own. And honestly, that is exactly how your body pulls off complex chemistry without needing a Bunsen burner. All right, let's dive into part 4, the real core of today's explainer, factors affecting enzyme action.
You have to consider this really delicate reality. Because enzymes are proteins made from these complex 3D folded structures, if you introduce extremes of heat, or say, you drastically alter the pH, you actually physically change the shape of that active site. And if the active site shape changes, well, the substrate just can't bind anymore. The enzyme is essentially broken. We call this process denaturation. And as you can probably guess, it is absolutely catastrophic for your metabolic pathways.
Because they are so incredibly sensitive to denaturation, literally all enzymes operate best under their own specific optimal conditions. For instance, the digestive enzyme amylase we mentioned earlier, it functions at absolute peak performance at exactly 37 degrees Celsius and a nice neutral pH of 7. If you drift too far away from those specific numbers, the reaction rate is going to drop and eventually the enzyme just denatures completely. Honestly, this is exactly why running a severe fever is so dangerous to the human body. You're literally risking the denaturation of the very enzymes that are keeping you alive.
Let's wrap things up with part 5, measuring the invisible. So how do we actually track all this microscopic activity in a lab? Well, one way is to measure the rate at which a product is formed. A really classic experiment for this uses our old friend catalase. Since we know catalase breaks down toxic hydrogen peroxide into water and oxygen, we can actually use simple potato cubes as a natural source of catalase. You just drop them into a flask of hydrogen peroxide, and you can literally trap and measure the volume of oxygen gas as it bubbles up into a measuring cylinder over a set period of time. Obviously, more oxygen in less time means you've got a faster rate of reaction.
Now, alternatively, instead of measuring a product appearing, we can flip it and measure a substrate disappearing. There's this brilliant little setup that uses a spotting tile to test amylase breaking down starch. We already know that iodine turns a dark blue-black color whenever starch is present. So what you do is mix amylase and starch in a test tube. Then, at regular time intervals, you pipette a single drop into a well containing iodine. Over time, as the amylase destroys that starch, the iodine will actually stop changing color. By timing exactly how long it takes for the starch to completely vanish, we can calculate the exact rate of reaction. And of course, you could easily repeat this whole thing at different temperatures to see exactly how heat affects the enzyme's speed. If scientists need even more precision, they'll use a tool called a colorimeter. This little machine shines a light directly through a sample to measure exactly how much light is being absorbed or transmitted. In our starch and amylase reaction, as the starch breaks down, the liquid actually changes color, meaning light transmission goes up. The protocol is pretty straightforward. You prepare serial dilutions of your stock solution so you have a nice range of known concentrations. But you really have to pay attention to step 1. You absolutely must calibrate the colorimeter with a blank, something like distilled water or a really weak iodine, before you start testing. If you forget to zero the machine, every single one of your high-tech readings is going to be completely inaccurate. Just a critical little lab tip to keep in mind.
Wow, okay, we've covered a massive amount of ground today. From the delicate induced fit hug of the active site, to the harsh realities of thermal denaturation, and finally how we actually capture this invisible data in the lab. But I want to leave you with this provocative thought. We know that extreme environments destroy enzymes, but if we perfectly understand the specific optimal conditions for different enzymes, how could we intentionally manipulate these environmental factors like heat, pH, or substrate concentration to perfectly control vital chemical reactions in things like biotechnology, drug development, or even mass food production? It turns out, mastering the invisible enzyme active site might just be the absolute key to mastering the physical world around us. Thanks for joining me on this explainer today, and as always, keep that curiosity focused.